Expasy ProtParam is great if you want to know the nitty-gritty about a protein, like its isoelectric point or pi, and its extinction coefficient that you can use to get its concentration from its UV absorption. And so Expasy ProtParam is a really useful tool, and so let's take a look.
So, bovine serum albumin or BSA is this protein that we often use as this like generic protein. And basically, it's just a protein that carries other proteins and molecules through the blood. You don't need to know much about it, but you can find it in Unipro, which I've talked about before, which is this database with a bunch of information about all of the proteins, all the species, etc. So if you were to look up bovine serum albumin, you would find that it has this accession number and this name. And now if you were to look this up in Expasy ProtParam, you would get information about it. So I'm going to enter the UniProt accession number. Oops, so I just reset it. I'm going to enter this and compute parameters. Now it's going to ask me to select which protein I want. So this protein gets processed and so you need to select the endpoints. So if you wanted to look at the sequence, you could go back and do that there. Otherwise, you can just use one of the predefined regions that it says. And so it's showing us that this is the main one that we would use. And so we click on that. We'll get more into later about how you can enter your own custom sequences, such as if you were to make your own shorter versions of a protein when you're doing some like recombinant protein expression, so making the proteins in the lab. Okay, so what this is going to tell you is that, so this is the sequence, and now it's going to tell you information about it. So the number of amino acids, this is just the length of the protein, the molecular weight, and so this is going to be in Daltons. Typically, we are talking in kilodaltons when we talk about proteins. So a kilodalton is 1,000 Daltons, and so this would be 66.4 kilodaltons, or 66.4 kDa. Okay, now it's going to tell us about the amino acid composition. So how many of each amino acid letter does it have? So proteins have combinations of these 28 common amino acids linked up in different orders, and this is why proteins are all unique. So proteins have different sequences and different numbers of each different amino acid, and this can influence some of the properties, as we'll talk about more in a bit. So it can be helpful to know the amino acid composition to see if there's any biases, things like that. So it'll give you the number of each of the different amino acids, as well as the percentage. And just to note that when amino acids link up to form proteins, they do so using their amino parts and their acid parts. And so the remainder, we call this remainder the residue. So each of the what was an amino acid, now these are referred to as the residues. Just a technical note that can get confusing. It'll also tell you about the total number of positively charged and negatively charged, and this will come in later when we're talking about the PI or the absorption. Oh, here we go. This is coming into play when we talk about the PI, so the isoelectric point. So here, as it's showing, it's this theoretical PI. So basically, the isoelectric point is going to be the point at which the protein is neutral overall. So as I mentioned, proteins have a different numbers of these different amino acids, and some of them are going to be. Some of these amino acids have the ability to either positively charged, we call those ones basic, or negatively charged, we call those ones acidic. And so the acidic ones, the ones that are often negative, are aspartate and glutamate, and the ones that are often positive, these basic ones are histidine, lysine, and arginine. And so if you were to look and you could then calculate like, okay, so how many of each of these does it have and what's the pKa of these? So basically at what pH are these going to be mostly neutral? Then you can calculate this thing called the PI, or the isoelectric point, which is the point at which the protein is going to be neutral overall. So much more on this in other posts, but basically pH is a measure of proton availability. And so protons are where the positive charge is going to come from. Is that these amino acids, some of them can get deprotonated or they're protonated. And then when these guys are protonated, they're positively charged. And when these guys are deprotonated, they're negatively charged. And if these were to be protonated, they'd be neutral. And if these were to be deprotonated, they'd be neutral. And whether or not they're protonated or deprotonated is going to depend on how many protons are around. And so If there are a lot of protons around, they're more likely to be protonated, and thus positive or neutral, depending on whether you're going from the basic, where they'd be positive, or for the acidic, where then they would be neutral. And how many protons there are around, we have a measure of that is the pH, which is the inverse log of the proton concentration. So the higher the concentration of protons, the more likely things are to be positively charged, and the more acidic the more base, sorry, the more acidic the solution. So the lower the pH. And so it's confusing because it's this inverse log scale. So the more protons you have, the more acidic, and the lower the pH. What's going to happen is that the pH is a measurement of the solution, the amount of protons in the solution. And then the proteins can give and take the protons. And they're more likely to take a proton if there's a lot of protons available, as well as the pKa of these individual ones. So how likely is this to take? How much does it want a proton? And how many protons are available? So those are the two things that come into play. You could do this for individual acids. At that level, you're talking about like the pKa. So at what pH does the proton availability and the desire to have a proton, when does that like balance out? And when do you have that halfway point where half of the molecules are protonated and half are deprotonated? And then if you're below the pKa, you're more likely to be protonated. And if you're above the pKa, you're more likely to be deprotonated. And what I'm talking about above or below, that's in terms of the pH of the solution. Now, what if you're talking about the protein overall? That's going to have a combination because it's going to come from the contributions of how many of these can be protonated and how many of these can be deprotonated. And so you can overall determine for a protein what the PI or the isoelectric point for the protein is, which is like the pKa of the protein overall. This is really useful because this PI can then be used to predict whether a protein is going to be positive or negatively charged overall, which can help us when we're trying to do purification based on a protein's charge. And so we can use this technique called ion exchange chromatography, where we can separate proteins based on their charge using little beads in these columns. We call this little beads resin. So we have these columns filled with this resin. It's either positive or negatively charged. And then we can flow our protein through. And so we want to make sure that our protein is going to be oppositely charged to the resin at the pH that we are using. And so we want to know, okay, is our protein going to be positive or negatively charged? And remember, this is overall, not there's still positively charged parts of the protein and negatively charged parts of the protein, but they overall balance out. Just a little technical note is that when you're doing ion exchange chromatography, it's actually like regions of the protein that are binding to the resin. So even if your protein is not fully, doesn't have a strong charge overall, it could still be binding. And so you can actually have your protein binding to both positive and negatively charged resin, but it's more likely to bind to the resin that's going to be opposite the charge that it is overall. And we can predict what the protein's charge will be by looking at its theoretical PI. This is telling me that BSA has a theoretical PI of 5.6, and this is theoretical because although you can calculate it based on those pKa's of the individual amino acids and how many of each they have, those pKa's are going to be determined when the amino acids are just like free in solution, and they're not considering that the amino acids are going to be like linked up into these chains, and the local environment is going to influence how much those amino acids want protons or don't want protons. And so it's going to influence the pKa of those individual amino acids and thus influence the PI of your protein. So it's a theoretical PI, but it's a good starting point. And so this is telling us that we have a theoretical PI of 5.6. And going back, this means that we're going to have the PI. So this is, imagine that we're talking PI here instead of PKAs. It's basically the same thing, but at the protein level. Now we are going to be at a pH that's typically, so think about if you have a pH of like 7.4 or whatever, so about the physiological or the pH in your body, you're going to be much higher than the pKa. So you're going to have fewer protons available. And if you have fewer protons available, fewer of the chains are going to be protonated. And if fewer of the chains are going to be protonated, your protein is more likely to be negatively charged. So we could say this is like an acidic protein. And if we were to look, we could see that it has 99 99 negatively charged residues and 82 positively charged residues. And I'm assuming that they're just determining this based on like whether they could be positive or negative. I'm not exactly sure. I think it just measures the As plus glute and the RH plus lyse, because these guys are going to be most likely to be positively charged, and these are going to be most likely to be negatively charged. When it comes to histidine, it's more wishy-washy. It's got this pKa that's closer to the to like physiological pH, so it can go back and forth more, whereas these are farther away, and so they're going to be way above or way below their pKa, and therefore they're likely to be protonated in the case of arginine and lysine, or deprotonated in the case of aspartame and glutamate. Okay, it also gives you some information about just like the atomic composition. So here we're looking at the amino acid level. Here we're looking at the actual atom level. So how many carbons, hydrogens, nitrogens, oxygens, and sulfurs does it have? As well as its like chemical formula, which I don't think is kind of funny because it's so ridiculously huge. 9,266 atoms. And if you do like X-ray crystallography, you're like, oh, that's a lot of atoms to map. But it's actually not that big of a protein, and as we saw, or again, it's kind of medium-ish, 66.4, but a lot of times people are doing structures with tons and tons of like complexes of so many atoms. But anyway, sorry, off track. Okay, the other really important thing that I use Expasy for is to get the extinction coefficient. So the extinction coefficient is going to let me get the concentration of the protein based on its UV absorption. So basically what happens is that there's this thing called the Beer-Lambert Law, and this says that if you were to shine light through a solution, there are going to be molecules in that solution that can capture some of the light. And this depends on the chemical makeup of the molecules. So different molecules absorb light of different wavelengths. And so because they're like absorbing or like stealing that light as it goes through, less of the you're going to get some of this light absorbed and less of it transmit or like go through transmitted. And so if you were to shine light through like a cuvette or through a nanodrop column that has the solution, molecules in the solution will steal some of the light, and you can measure how much got absorbed by measuring how much you put in and how much you got out. And then if you know, Beer's law says that this is directly relational. The amount that's absorbed is going to be indirectly related to the amount of the light stealer, so the concentration of the molecules that can absorb the light. And different molecules will absorb the light of different wavelengths to different amounts. And this is taken into account with this thing called the extinction coefficient, which is going to tell you how well the molecule absorbs that wavelength. I'm telling you this because proteins absorb UV light, so this light in this ultraviolet range. So it's a higher frequency than visible light, but it's like the same type of, exact same type. It's just on the spectrum. It's just higher in frequency than visible light, but it's the same type of it's the same little photons, packets of energy traveling in waves, whole thing above. But anyway, so proteins, different regions of proteins will absorb UV light, and in particular, these aromatic amino acids. So these ones with like these resonance stabilized rings. Basically, there's this thing called electron delocalization, where all of these guys are kind of sharing their electrons, and so each of these lines represents a bond. And bonds that involve sharing of electrons, and here they're sharing electrons among like all of these. And so you have this thing called resonance or conjugation, various terms. But anyway, this makes it more likely to absorb this UV light. And so we can see that the aromatic amino acids are going to absorb light at around 280 nanometers wavelength. Especially tryptophan. And so the amount of tryptophan you have, the more tryptophan you have, the more your protein is going to absorb, as well as these absorb to a lesser extent, and you also get absorption from the protein backbone at like 230. But anyway, we're typically measuring at 280 because that's going to give us the most specific information about the protein, as opposed to like nucleic acids or other contaminants. And so And this is going to be directly related to how many of these aromatic amino acids we have. So you can imagine if you had a lot of tryptophan, then you would absorb more. So you would get tricked into thinking, oh, I have a lot of protein, when really you have less of the protein. Your protein just happens to have a lot of tryptophan. So it's important that we know the extinction coefficient for each different, for the protein that we're studying. And this is assuming that we have like a purified protein. So if we don't, and this comes to another important point, is that we can think of like a generic protein, that we could use an extinction coefficient for like a generic protein. And so often you'll be doing that. But if you have because you won't know if you have a combination of different proteins what the extinction coefficient for the combination is. But if you know, for like a pure protein, then you can use this Beer-Lambert law with the PI that's calculated based in this uniprot, this ProtParam tool. So here it's telling you the extinction coefficient. So I could plug this into Beer-Lambert law and get the concentration based on the absorbent on the path length, which this is typically going to be like automated by your machine. And there's also this thing like this absorption 0.1% equals 1 gram per liter. So basically 10 grams per liter is 1% weight volume. So 0.1% is going to be 1 gram per liter. It's just like a convention. So this is saying that if you're reading an absorption of 0.646, this is going to correspond to your protein being at a concentration of 1 grams per liter. So say you had a concentration of 0.323, then you would have half a gram per liter, et cetera, et cetera. But this is assuming that all the pairs of cis residues form cysteines, and this is probably not likely what's actually the case in your case, because typically you're using reducing conditions. So much more on this in other posts, but basically this amino acid cysteine can get oxidized and form these crosslinks, these cysteine crosslinks, these sort of disulfide bonds. This can be helpful for proteins that are like secreted and outside of the blood. So maybe these typically are actually in the cysteine form. If these in BSA, I'm not exactly sure. But because it is like out in the blood and stuff, and these bonds can be good for helping stratigraphy the protein. But typically for like intracellular proteins, they will be under reducing conditions, and often, and typically when you're working with proteins, you're going to be under reducing conditions because you don't want these like crosslinks to form inappropriately between different proteins or between regions of the protein, but just like locking the protein into weird positions. And so we typically use reducing agents like DDT or beta-mercaptan, more in other posts. But anyway, so you have this, so this is assuming that those are all going to be oxidized. You can see that if you under reducing conditions, you're going to have a slightly different extinction coefficient. So typically this is the one that you're going to be using. Okay, a couple other things. There are other ways to measure protein concentration as well. And so I have a post on that as well, different ways you can measure things. When it comes to a common one is this Bradford method, where you use a dye that binds to proteins, and then you measure the amount of blueness, because the dye is going to change to this blue color when it binds to the protein. But note that this one is going to be biased by basic letters, so lysine, arginine, histidine, as well as things like tryptophan, phenylalanine, and tyrosine. So it gets biased by these aromatics as well. So what happens is that Bradford assay, you're going to actually have BSA is not that great of a protein for using as a standard, despite the fact that we often use it as a standard. Because if you were to do like a standard curve, you can see that it has a much higher absorption than other proteins. And so each protein is going to have differences. And so although we typically use like BSA or BSG or whatever to try to estimate like a total protein, it gets complicated when you have a mixture of proteins, which is why if you have a pure protein, it's really nice to be able to use this extinction coefficient. Okay, just a couple final things that are on here. One is the estimated half-life. So I'm not actually sure I, why you would or when this would be, how this is calculated. I mean, but this is the estimated half-life if you are interested, or I guess it's measured in vitro. Yeah, I'm not exactly sure. I guess they measure it in cells and then tell you. Okay, so but basically this would last about an hour in a million reticulocytes. Instability index. Okay, I've almost always had this tell me that my protein is considered unstable, but it has an instability index of this. Aliphatic index. So aliphatic is basically the opposite of it. Anything that's not aromatic is considered aliphatic. So things that aren't aromatic will be aliphatic. I'm not sure what this aliphatic index is though. Grand average of hydrophobicity or gravy. So basically um, amino acids that are hydrophobic will be like excluded by water. This will make the proteins kind of like, those will make those amino acids clump up inside of the protein, and it'll influence how the protein folds. If those are on the surface, then like it will also, it can maybe make a membrane bound protein, various things like that. But a protein that's more hydrophobic is going to be more excluded by water, often, and this could influence things and where it's located in cells, et cetera, et cetera. And this is going to use this gravy score to tell you the grand average of hydrophobicity, which I'm not exactly sure how to interpret the numbers, but something that you could look into if you were interested in. Another thing, okay, so we were using, based on the accession code, we could also, once we saw that we could also determine, select the endpoints on the sequence, or use a different and then see terminal. We could also just copy and paste our own sequence in here. And this can be helpful if you're doing like recombinant protein expression. So if you're making, expressing this protein in the lab, so taking the genetic instructions for making it, sticking it in cells and getting the cells to make it. And since you can manipulate the sequence, you can make changes to see like what different regions the proteins do, et cetera. And you can make these changes using methods like SLIC or a quick change directed, it's like directed mutagenesis. So there's various ways that you can make different mutations to the protein and see what they do. When you make mutations to the protein, if you're chopping off a region, maybe see what that region does, or you're just testing the region alone, or you're making a point mutation. All of these changes can influence the proteins. PI can influence the proteins. Extinction coefficient, also if you're adding a tag, that will influence things. And so you're going to have these like custom protein constructs. So basically just different versions of the protein that you've made, and you want to know information about those proteins. And so you can then paste that sequence into here and get the information for that, the one that you're using, because remember that's going to influence the extinction coefficient, the PI, etc. So that's the basics of using Expasy ProtParam, and I hope you find it helpful.